Hello everyone, welcome back to my channel, the Zoology World. In today's video, we are going to talk about the different cell organelles. The first organelle is endoplasmic reticulum. It is also known as ergastoplasm or cell circulatory system. It's a system of membrane-lined channels found in all eukaryotic cells, and it is named as sarcoplasmic reticulum in skeletal muscles, nasal granules in neurons, and myeloid bodies in retinal pigmented cells. The endoplasmic reticulum has three parts, as you can see in this diagram. The cisternae, tubules, and vesicles. The cisternae are the sac-like, unbranched, parallel tubules having ribosomes on their structures. The tubules are tube-like, irregular, branched, without ribosomes. And the vesicles are round or spherical with or without the ribosomes. The ribosomes can be of two types, as you can see in the first diagram. They can be rough endoplasmic reticulum and smooth endoplasmic reticulum. Firstly, I am going to talk about the rough endoplasmic reticulum. They have ribosomes. 68 ribosome units are attached to the ER by means of the protein riboforin 1 and riboforin 2. The number of cisternae and tubules are more in the RER. They are rich in protein secreting cells, example, plasma cells, goblet cells, fibroblast cells, pancreatic cells, and liver cells. They are abundant near the nuclear membrane. Now, let's talk about the smooth endoplasmic reticulum. They are devoid of ribosomes. The number of tubules and vesicles are more. They are more near the plasma membrane and Golgi complex. They are rich in lipid and sterol secreting cells, example adipose tissue, interstitial cells, muscle cells, liver cells, etc. They are also synthesize the carbohydrates known as the glycosomes, vitamin C, and helps in the detoxification of pollutants and carcinogens done by the enzymes P450 and P448. The smooth endoplasmic reticulum is also known as agranular, and rough endoplasmic reticulum also known as the granular. They are absent in the mature RBCs, egg cell, and the embryonic cells. They arise from the evagination of the nuclear membrane. There are 30 different types of enzymes found in the endoplasmic reticulum. The functions of the endoplasmic reticulum include providing mechanical support to the cell, intracellular transport from rough endoplasmic reticulum to smooth ER, then to Golgi bodies, then to primary lysosomes, and then out of the cell by exoosmosis. There are many enzymes in the endoplasmic reticulum that helps in the metabolic activities of the cell. It also helps in exchange of the material between the cell and the external environment, and it also helps in the formation of primary lysosomes and synthesis of the nuclear envelope. The next organelle is Golgi complex or Golgi body. It is also known as Dalton complex or lipochondria or idosomes. It is present in all eukaryotic cells, but it is absent in the mature RBCs. It is surrounded by a zone of exclusion. It is the area that is clear region where ribosomes, mitochondria, and storage granules are absent. It originates from the ER. It exists in two forms. The localized form known as the Golgisome. It have a fixed position near nucleus, example in vertebral cells, and second one is the diffused, isolated or dictyosomes. It is scattered in the cytoplasm. As you can see in the diagram, this is the dictyosome, a scattered form of Golgi body that is commonly found in the plant cells. The Golgi apparatus have definite polarity. It have distal, maturing or trans phase that is concave in shape and it is facing toward the plasma membrane. The proximal, forming or cis phase is convex in shape. It receives material from the ER and cytosol in the form of transitional vesicles. The Golgi apparatus has three parts like that of the ER. It has cisternae. These are sac-like, unbranched 3 to 12 parallel or concentric tubules. Second are the tubules. They are tube-like, branched more towards the forming phase. And the vacuoles. These are sac-like, present near the maturing phase. The main enzyme found in the Golgi body is nucleoside diphosphatase. The functions of the Golgi body includes: it helps in the intracellular transport system. It modifies the materials like proteins, carbohydrates, sterols, etc. and store them. The Golgi body forms secreting vesicles that discharge their contents to the exterior, for example, tears, sweat, saliva, and hormones. They carry out the membrane transformation. It helps in formation of lysosomes. It forms the acrosomes in the sperm. They carry out recycling of the plasma membrane. The production of hormones in some of the endocrine glands is mediated by the Golgi apparatus. For example, thyroxine hormone in thyroid gland. Golgi complex also produce pigment granules in tumor or cancer cells. Next is the lysosome. The lysosome, as you can see here, it shows polymorphism. It exists in different forms. First form is primary lysosome, also known as the storage granule. It is a vesicle that is filled with inactive hydrolytic enzymes. Then is the secondary lysosome or heterophagosome or simply phagosome. It is a lysosome that is fused with the ingested food. Third is the residual body or tertiary lysosome. It is a lysosome left with only undigested food, which is later thrown out by exocytosis. And the fourth form is the autolysosome or autophagosome. It is a primary lysosome feeding on the cell organelles or worn out cells. The lysosome, it consists of a membrane which contains acidic hydrolytic enzymes in the form of minute crystalline or semi-crystalline granules. They are found in cells which are involved in the intracellular digestion. For example, WBCs in blood, phagocytes in liver, histocytes in connective tissue, and osteocytes in bones. There are 40 to 50 different types of these hydrolytic enzymes. They include nucleases for digestion of nucleic acid, lipases for lipids, phosphatases for phosphate-linked compounds, sulfatases for sulfate-linked compounds, and glycosidases for polysaccharide digestion. The lysosomes are synthesized or arised from the Golgi complex or smooth endoplasmic reticulum. The different functions of the lysosomes include intracellular digestion. It also includes in the extracellular digestion, as in case of the acrosome. It produces hyaluronidase to digest the egg envelope. They also digest the harmful substances and provide defense to the body. They are also involved in release of hormones, for example thyroxine from thyroglobulin. During metamorphosis, they help in digestion of unwanted organelles, for example tail in case of the larvae or tadpole of the frog. During starvation, they hydrolyze the stored organic food of the cell and provide extra nourishment. They cause the breakdown of nucleic acid that results in mutation. Some of the residual bodies, when accumulated in the cell, they cause storage diseases like gout. They can reduce aging. For example, rotting of fruit is reduced by checking the action of polygalacturonase. Next organelle is the ribosome. The ribosomes has two parts. Smaller one is cap-like and larger one is dome-shaped. The ribosome is defined on the basis of their sedimentation or Sedberg unit, also known as Dalton unit, that is equal to 1 into 10 raised to power 13 second. The prokaryotic ribosomes and the organelle ribosomes in eukaryote is 70S, which has 50S and 30S subunit. The eukaryotic ribosome has 80S, that is 60S plus 40S. Each unit of the ribosome is composed of the rRNA and protein complex, so they are known as the ribonucleoprotein. The composition is 70S, 50S subunit has rRNA of 23S and 5S and proteins 34 types. The 30S subunit has 16S rRNA and proteins 21 types. So the RNA to protein ratio in prokaryotes is 60:40. And in case of the eukaryotes, the 80S subunit, larger subunit 60S, has rRNA of 28S, 5.8S and 5S with 45 types of proteins, and the smaller subunit 40S has 18S rRNA with 33 types of proteins. So the RNA to protein ratio in eukaryotes is 50:40. The ribosomes are the smallest organelles. They are non-membranous. Both eukaryotes and prokaryotes have them. In prokaryote, it is generally found in the cytoplasm. So there we call it as cytoribosome. In eukaryotes, the organelle ribosomes, like in plastids, known as plastidoribosome, and in mitochondria, mitoribosome. Magnesium 2 plus ion is required for the binding of the two subunits. The larger subunit has a A site known as the acceptor site, but the P site known as the peptidyl site, it is formed by both the subunits. There are many ribosomes which are attached to the same mRNA, and the complex formed is known as the polyribosome or polysome or ergosome. It is more common in prokaryotes as a similar type of protein as to be found there. The functions of the ribosomes include protein synthesis, and it helps for providing the site for attachment and also providing the protection for the mRNA and tRNA. It also protects the nascent polypeptide. Next is the mitochondria. It is a double membrane structure. The outer membrane is permeable and it is stretched tightly, and the inner membrane is selectively permeable. It forms the folds known as cristae with large number of bat-like structures attached to it known as oxysomes. These oxysomes, also called F-node F1 particles, they are meant for oxidative phosphorylation. It is the second largest organelle. 70% cells enzymes are found in the mitochondria only. It is a self-replicating organelle as it consists of its own DNA. The lifespan of the mitochondria is about 7 to 10 days. It is absent in mature RBCs and anaerobic cells. The number of mitochondria found in a cell depends upon its metabolic activity. For example, kidney has about 300 to 400 mitochondria, oocytes have 30,000, and liver cell has 1000 to 1600 mitochondria, and in yeast it is less than 10. The functions of mitochondria include cellular respiration. The outer chamber of the mitochondria help in elongation of the fatty acid chain. The yolk formation during the ovum development, it is done by mitochondria. The mitochondria store and release calcium 2 plus ions whenever required. It is a site for thermogenesis, that means heat development. And the middle piece of the sperm, which provide the energy for the movement of the sperm, it is also formed by the mitochondria. The vacuoles. The vacuoles are fluid filled, that is known as the sap. They are the sap sacs that are covered by a semi-permeable membrane known as the tonoplast. They arise from the ER and Golgi complex. The animal cell has small sized vacuoles which are larger in number. In plant cell, there are large size vacuoles which are small in number. The cell sap contains water, minerals, gases, sugars, organic acids, wastes, and pigments. These vacuoles can be of different types like sap vacuoles as found in plants, contractile vacuoles, food vacuoles, and gas vacuoles. The functions of these vacuoles include: vacuoles keep the plant cell turgid. Sap and contractile vacuoles are made for the osmoregulation. Vacuoles contain pigments which attract birds and insects for pollination. Food vacuoles store minerals, carbohydrates, etc. And the pigments in the vacuoles provide color to flowers, roots, and stems. They also store the waste products and plant metabolites which discourage herbivores from eating the plants. Next are the cytoskeleton structures. They are visible under the electron microscopes. These components can disassemble, move to a new location, and can reform as needed. The structure includes three types: microtubules, microfilaments, and intermediate filaments. The microtubules are hollow cylinders. These are contractile structures. They are formed by the tubulin protein. Calcium 2 plus ion, magnesium 2 plus ion, GTP, and calmodulin are required for assembling of the microtubule subunit. They are present in all cell matrix except in slime molds and amoeba. Their number increases during the cell division. The functions of these microtubules include: they help in providing shape and support to the cell. They help in movement of macromolecules and cell organelles, so they form the microcirculatory system. They form the spindles during the cell division. They form the motile elements like cilia and flagella. They are also the components of the centriole and basal bodies. They play an important role in determining the polarity of the cell. The microfilaments. These are the solid, rod-like structures. They are made up of two types of proteins: the globular protein known as actin or microfilament, and the fibrillar protein known as myosin or macrofilament. Their subunits or monomers are contractile in nature. Their functions include supporting the matrix. They help in cyclosis or cytoplasmic streaming movement. They also help in exocytosis and endocytosis of the plasma membrane. They form units together for the muscle contraction. They also help in cleaving of the animal cell. The intermediate filaments are solid, non-contractile, and made up of 18 protofilaments. These are larger than the microfilaments. They are absent in the mature RBCs. They are made up of different types of proteins like keratin, desmin, and viminatin. They maintain the cell shape. Desmin and tonofibrils are forming the desmosome structures which form the interconnections between the different cells. Keratin is found in our hairs. It is the structural protein there. The neurofibrils in the axons in the central nervous system, they help in providing the impulse conduction. Next is the centriole. The centrioles are hollow cylinder filled with matrix. Their walls are made up of 9 microtubule triplets arranged in a ring. This arrangement is called 9+0 pattern. Inside the microtubules, a cartwheel structure is formed. Each centriole is surrounded by a dense sphere called the crown or pericentriolar satellite, which keeps on changing their position. They help in assembling new centriole in genode phase. These are non-membranous, self-replicating structures. These are present in pairs, so called as diplosomes, surrounded by a distinct cytoplasm known as centrosphere or kyanoplasm, and these together form the centrosome. It has a high amount of ATPase enzyme. It helps in spindle formation and astral rays formation. They form the basal bodies of the cilia and flagella, and the distal centriole of the sperm form the axial filament. Cilia and flagella. These are semi-autonomous organelles. That means they have some degree of self-government or self-regulation. They arise from the basal bodies. These are found in ciliates like protozoans, flame cells of the flatworms, epithelial linings in the respiratory tract, oviduct, renal tubules, choanocytes in sponges, gastroderm cells in coelentrates, etc. They have a double flagellar sheath. The flagellin protein form the cilia and flagella. Their functions include they help in locomotion, they help in capturing the food, they help in removal of the waste from the respiratory tract, they help in delivery of urine and gametes from the oviduct and urinary tract. In some cases, these are also sensory in nature. As you can see in this diagram, the flagella has 9+2 arrangement and it has a double outer sheath. The cilia, it also has the same 9+2 arrangement with an outer sheath. The basal body, which are forming the base point of the cilia and flagella, they can change to centriole. They have the same structure as that of the centriole, that is 9+0 pattern, but they can occur in eukaryotic cell which bear cilia and flagella and are without the centrosphere. The basal plate. It is a plate-like band present between the basal body and the shaft of the cilia or flagella. In this, the C subunit fibers disappear and additional central singlet fibers appear. The last organelle we are going to discuss is peroxisome. These are membrane-bound microbodies consisting of crystalline core surrounded with a dense matrix. They contain enzymes for peroxide biosynthesis, that is, producing and destroying the peroxides. The functions of the peroxisomes include they play important role in the metabolism of sugars, amino acids and fats. Peroxisomes detoxify the alcohol in the liver by transferring hydrogen ion H+ to oxygen. That's all about the cell organelles. If you find this video easy and informative, don't forget to like and please subscribe my channel. Thank you.